Start. So, welcome everyone for the CA2 of Wenjong Shon, and your supervisor, principal supervisor is Professor Ho Kyung Shon, and co-supervisor is Associate Professor Sheru Punsho. And as your technical assessors for today, it will be Dr. Yang Woo Chu and Associate Professor Leonard Tihing. Wenjung, you have 15 minutes. This is a CA2, so it's a closed session. You can start now.
Yeah. Good afternoon, everyone. I'm Wenjung Son, and today, as my candidature assessment stage 2, I'd like to share my current research progress and outcomes. Yeah. Firstly, I'd like to briefly introduce my research topic, which is compact and high-performance activated carbon incorporated membrane bioreactor to transform urine to fertilizer for a circular economy. My main focus is ultrafiltration membrane bioreactor with powdered activated carbon for effective nitrification of urine, which enables complete nutrient recovery from source-separated urine as an NPK liquid fertilizer. However, a major challenge in operating urine treating MBR is long hydraulic retention time, which can result in a large footprint and more energy consumption. As such, my research aim is how to improve this challenge of long HRT. The structure of my PhD thesis has been developed under the guidance of my supervisor, and my thesis will consist of 8 chapters, and chapter 4 to 7 will be the core of the thesis, which are technical studies. Based on these chapters, now I'd like to share my current research progress, critical findings, as well as study plans. My first technical chapter focused on the effect of temperature and dissolved oxygen on the nitrification in a membrane bioreactor treating source-separated urine. Although temperature and dissolved oxygen are important operational factors, which are highly related to nitrification performance, previous studies have only been conducted at the room temperature. Therefore, this study investigated the effect of 3 different DO levels with 3 different temperature conditions, varied from 10 degrees Celsius to 30 degrees Celsius. The result is summarized in the table. And as you can see, 20 degrees of temperature with 4 milligram per liter of DO showed the best nitrification rate of around 201 milligram per liter per day, and the following short HRT of around 8 days, as well as better TOC removal rates compared to other conditions. This experiment of this study has been finished. However, microbial community analysis is currently under planning for further in-depth supporting information. Chapter 5 focuses on the membrane bioreactor incorporated with biofilm carriers and activated carbon for enhanced nitrification of urine. As can be seen in the schematic diagram, biofilm carriers were added to PAC-MBR to support more biomass, especially slow-growing nitrifying bacteria, leading to HRT reduction, compact and high-performance MBR system. The result showed a 36% increased nitrification rate and thereby 40% HRT reduction and 11% increased TOC removal rate. Microbial community analysis result showed that the predominant AOB genus was Nitrosococcaceae and NOB genus was Nitrospira. The predominant AOB Nitrosococcaceae showed 7-fold higher enrichment by attached growth on the biofilm carriers rather than suspended growth. So overall, the incorporation of PAC and biofilm carriers significantly enriched the abundance of nitrifying bacteria compared to the conventional MBR. In Chapter 6, hybrid MBR and membrane aerated biofilm reactor system for nitrification enhancement will be included. And this study is currently at the initial stage of setting on experiment plans. So to briefly introduce the concept of the study, bubble diffuser, normally used for aeration in MBR system, has low oxygen transfer efficiency as well as high energy demand. On the other hand, membrane aerated biofilm reactor, MABR, utilize hollow fiber membrane as an aerator to diffuse oxygen through the membrane to the attached biofilm. So this MABR features high oxygen transfer efficiency, thereby saves energy and highly enhances nitrification. However, due to the anoxic zone in the outer layer of the biofilm, denitrification can easily happen under high COD concentrations. As such, this study will focus on the initial COD remover in MBR system and the following MABR system for effective and faster nitrification. And my last technical chapter will focus on the compact pilot scale MBR system. And this study will focus on the effect of different fixed HRTs on the ammonia to nitrate conversion ratio. And by applying the produced fertilizers into a hydroponic plant growth, this study will be able to examine the threshold of HRT in the MBR and conversion rate to be an effective urban fertilizer. Now I'd like to share some research outcomes since my CA1. In terms of journal publications, based on my literature review, I published one review paper, and based on the Chapter 5 study, I also published one technical paper. And another manuscript focused on the microbial analysis of Chapter 5 study has been submitted to Bioresource Technology, and now I'm addressing the reviewer's comments. And I also published 3 papers as a co-author. And since my CA1, I actively attended 2 international conferences for oral presentation, such as Richard Summit 2022 and 2023, and IMSTEC 2022, and CESE conference in 2023. And this chart is showing my whole PhD study plans and progress. So firstly, for Chapter 4 study, the experiment of operating MBR has been completed, but microbial analysis is remaining at the moment. And for Chapter 5, the experiment has finished and published 2 papers. And for Chapter 6 study, the experiment will be started in the near future after careful planning. And for the last chapter of pilot MBR study, I started the operation and now acclimating the sludge. So after the operation, the hydroponic application as well as microbial analysis will also be carried out. At the same time, thesis preparation and candidature assessment 3 will be completed in my last PhD semester. Yeah, thank you.
Thank you, Renju. You kept to the time and your presentation was clear. Okay, I will hold my comments. I'll start first with the SSs. Dr. Yang, you want to start first?
Yes, thanks, Gayatri, and thanks, Wanjung, for the presentation. Seems like you developed from your CA1. I was also a panel on the CA1, as I recall, and then seems like the thesis has been developed well, and your chapters are coherently connected, which is, I think it's positive. I have a few technical questions about the about your research progress. So can you go back to the chapter 4 results? So yeah, it's just out of curiosity, how did you maintain the temperature to 10 degrees C? It's a sub room temperature.
Yeah.
And yeah, can you briefly explain?
Yeah, for the temperature maintenance and adjustment, I use the circulator, as you can see in this schematic diagram. And the reactors has double jacketed, double layered glass reactor. So by circulating the water outside of the reactor, and I could maintain the temperature to 10 degrees and 20 degrees and 30 degrees.
Okay, I see. And your results seems like it's not linearly increasing or decreasing as a function of the temperature and as well as the oxygen contents. And as I read your report, you briefly explained about why, but it was not too clear to me. So can you explain why it's peaking at, for example, like a 20 degrees C rather than it's monotonically increase as a function of temperature?
So temperature is very important operational parameters for nitrifying bacteria for nitrification rate. So initially, like other previous studies showed 10 degrees Celsius maybe has higher nitrification rate than room temperature of 20 degrees. And 30 degrees Celsius can be sometimes higher nitrification than the room temperature. So that's why I planned this temperature variations to see how the nitrification rates and HRT can affect the nitrification rate of this urine treating MBR. And my result is showing that 20 degrees had the best nitrification and shortest HRT rather than 10 degrees Celsius and 30 degrees Celsius. It means in the 30 degrees Celsius, the AOB and NOB balance, AOB nitrifying bacteria and NOB bacteria balance may be affected by the temperature. So that's why nitrification can be affected. So initially I was maybe expecting, my research question was maybe in the 30 degrees nitrification can be more active and nitrification rate should be higher or HRT can be reduced. But from my experiment, in the high temperature of 30 degrees, actually nitrification decreased.
Yeah, that was actually my point. So I'm not the expert on the microbial activities, but yeah. In general, yeah.
I'm assuming that the AOB was growing faster than the NOB in 30 degrees. So nitrite accumulation happens in 30 degrees. So that's why the nitrification and HRT affected in my experiment. So I'm planning to do the microbial analysis of sludge samples that results can explain more of this result, I think. So I'm planning, more analysis of this result.
And as I see your temperature versus days data, so it seems like you sampled the 10 degrees C sample in the earlier days and the 20 degrees C samples in the middle days and the 30 degrees C sample in the later days. And if your MBR reactor has some hysteresis or if it has some history effect, then it may also affect your results. So I think you probably rigorously check this history effect if there's any sort of like this sequence has affected your results. So yeah, that was just a comment. Yeah.
Yep,
Right.
thank you.
Yeah. And regarding on the chapter 6, you are trying to aerate the solution. And my question is that so you also already studied about the dissolved oxygen, and what's the fundamental difference between the aerating and the changing your dissolved oxygen concentration? What's the main difference between this one versus the chapter 4 study?
Oh, so chapter 4, in the chapter 4, I just used the normal bubble diffuser, as you can see in this picture for MBR. So it has large bubbles, coarse bubble and fine bubbles. So it can also affect the sludge formation, floc formation, and it can also affect nitrification of MBR system. But I'm trying to apply this MABR, which is the aerator is from the membrane, hollow fiber membrane. So it doesn't have very large bubbles. It just diffuse oxygen through the membrane and trying to form the biofilm on the membrane. So it has higher oxygen transfer efficiency. So in this case, so dissolved oxygen can be controlled by the pressure of compressed air through the membrane. But with the small, maybe energy can be saved more compared to the MBR. And so you mean the difference of dissolved oxygen concentration control in the chapter 4 and this MABR system DO concentration, right?
Yeah.
I'm actually in the initial stage of figuring out the experimental plans of this MABR. So as far as from my literature review, this MABR system it depends on the formation of the biofilm. So if it has a high diffusion, then biofilm cannot form on the membrane. And if it's too small, then biofilm cannot also be formed. So controlling the pressure of air through the membrane is the key factor in this case, not the dissolved oxygen.
Okay.
Yeah, so I'm trying to see how the biofilm can be formed on the membrane first, and then maybe controlling the biofilm thickness of the biofilm can be the critical factor for the best nitrification.
I see.
Yes.
Okay. So it's a different perspective. Okay. I understand. Okay. All right. Yeah, that's all.
Thanks, Yang-Woo. Now to you, Lennon.
Yeah, thank you very much, Gayatri, and thank you, Wanjung, for a nice presentation. I thought overall you did very well since your CA1 report. You performed well. You have produced and published 2 papers, I think, and a few more as co-author. And you have presented in many conferences as well as forums. So that's really good of you. I have a similar question with Yongwoo actually on the effect of the temperature. But I was just wondering, you said about AOB, NOB balance. That's one of major factor on that one. Have you checked on that measurement so far to support your whatever theory you have on that difference in the
regarding the AOB and NOB like communities and abundance of the sludge, I'm planning on the microbial analysis for this chapter 4. Yes. So For now, I'm assuming from the literature review, but I will analyze the microbial community as well to support my result.
Okay, is this ongoing now?
Yes, it's ongoing. Yeah.
Okay, that's fine. May I revisit some of the questions or comments from your CA1 and see how you progress from there? I'm just looking at your CA1 comments right now. There's a comment here about to recheck some technical details on your nitrification over HRT rate results. Have you done, and what's the update on this one? Have you done any further analysis on this?
Yes, from my CA1, I got comments that clearly investigate the nitrification rate and HRT.
Yeah.
So it was Dr. Sharup's comments.
So this is
And
the one that you're showing now, yeah?
Yes. So
Okay.
nitrification and HRT is closely related. So nitrification rate, HR depends on, no, the HRT depends on the nitrification rate in the MBR. So that's why I'm focusing on the nitrification rate and HRT as a subsequent, as a following factor. So I'm focusing on two factors to see the close relations.
Okay,
Yeah.
thank you. So in your process, you're using MBR, you would expect some fouling issues as well. I think we discussed this one in CA1 as well. Is there any major fouling issues in this process, or have, of course, the aeration probably would help in reducing that one, but any major fouling issues?
In terms of membrane fouling issues, we can't really see deterioration of membrane performance because we are only keeping the low LMH flux, so around 3 to 5 LMH. So it's very slow reaction in the MBR, as you can tell from the HRT, long HRT. So I cannot really find any membrane fouling issues at the moment.
Okay, that's fine.
So far, yeah.
Okay, can you go back to your timeline, please? I think this is just the last question that I have. All right, so the last few remaining ones you're working on is chapter 6, which, oh, you have, when is that, 2024? It's not started yet, right? So
Not started yet, it will be started soon after, yeah, planning.
So you're working on the pilot scale right now?
Yes.
And also the microbial analysis in chapter 4.
Yeah.
Right. So I'm just looking at your timeline. So you have one more year to go, and some of your chapters will be done in concurrently. So yeah, okay. So they should not have any issues. Do you have any issues with your pilot testing so far, or everything's okay?
Yeah, we newly developed this compact pilot system, MBR system. So yeah, definitely we have some small issues, but I'm figuring out how to solve the problems and how to operate the pilot scale MBR system. Right now, I'm in the middle of figuring out the pilot system as well as acclimating the sludge.
Okay.
So it's in this starting up stage of this system. So my experimental plan will be started soon. So hopefully I can make all things done in the timeline.
Okay. Yeah, overall, I think you did very well in terms of the storyline. It looks like there's a good flow of the different chapters there. Of course, I would like to see as well on the result of that MABR system, as you are saying, that is more less costly. It's less costly compared to the normal aeration part. So that needs to be seen, and how the performance of that band. But in terms of flow and the direction, I'm okay with that. So congratulations, Wanjung. Thank you.
Thank you.
Thanks, Gayatri.
Thanks, Leonard. Now we move over to the co-supervisor first. Professor Sheriff, for your comments.
Yeah, thank you, Wonjong. I think, yeah, I agree with Leonard and Young that your presentation was quite good. I think only you didn't mention about what how you have addressed from the CA1. I think this is what many even previous. I think yesterday also was lacking. So I think that's what I think one of the our students need to know. I think can you go back to your slide in where you have shown the layout for your experiment MBR? Back. Yeah, okay, this one. Oh, next one. Yeah. So when you present this one, okay, so when you present like this, see what R1, R2, R3, what does it mean? Does it mean MBR1?
Oh, sorry
and
for
MBR2?
this. Yeah, R1 is MBR1
Yeah.
and MBR2, MBR3.
Yeah, so
Yeah,
I think
I didn't change.
Yeah, try to maintain similar because it looks like R1, R2 are a little bit different. So here I was just wondering why, because you said that, so your probable cause for the reduction in nitrification at 30 degrees Celsius is likely because of the nitrite accumulation, right?
Yes, as well as maybe inactivation of nitrifying bacteria.
Right?
Yes.
So I think you should have presented here the data for nitrite concentration. So did you tested that, right?
Yes.
So how was your data for the nitrite concentration? Is it accumulating or not? How does it compare for?
Yeah, in 30 degrees nitrite also accumulated in all three reactors. But in the highest DO level accumulation concentration was highest among these three reactors. I didn't include that graphs because due to the time limitation in this CA2, but yeah, it will be in the thesis and manuscript after I complete the microbial analysis. Yeah.
So I think that those are sort of things that you need to present because that was your main explanation to support that one. No?
Yeah.
So I think you should have presented that data. I think that's all I have. I don't have. Overall, I think the progress is quite good. I think 2 more remaining, and most of the works have been done. So MABR and pilot, I think pilot should be fine because it's been already started. So how is your progress with MABR?
MABR progress is now we are trying to figure out how to put the MABR membranes in a module. So after we solve that problem, maybe the progress will be faster, I think. Yeah.
So you have to submit your thesis by the end of this year, right?
No, end of April in 2025. So next year, April.
Okay, I'm good. Yeah, thank you.
Thank you, Cheryl. Wenjung, can I have a look at your timeline slide, please, and also your conference that you attended? This is fine. For CS, you also attended you also took part in the research showcase and you won, right?
Yeah.
Yeah. Next time, please include that because that's a big achievement, right?
Okay.
Unless you don't consider it a big achievement, but it is. So you should. It is
Yes.
a research outcome. Yeah. Okay. And okay. So for your timeline, if kindly go to your timeline, please. So you do have. I mean, you've covered quite a substantial amount of work, so you do have a good time. And so this time is especially focused towards operating the pilot scale system is what I'm assuming that your primary goal here. Do you have like a particular amount of time, like you need to run the pilot scale system that you aim to run the pilot scale system?
I'll continuously operate this pilot system, but for my objectives, research objective for chapter 7, maybe the one set of experiment will be completed in like 3 to 4 months. But after that, I'll continuously just operate this pilot system. And at the same time, I'll do the chapter 6 MABR study. Yeah,
Okay.
I'm not gonna finish this pilot system because once I started I wanna keep the sludge, and the pilot system is very important for our whole project. So
That's right. Okay,
yeah.
so my question is just wondering, you have done all the experiments from your chapter 4 to 6 using lab scale system.
Yes,
So of
that
course in
was
lab
only
scale
that.
system, you know, when you explain to Dr. Yang Wu how you maintain the temperature and how that you maintain the bacteria and the of course the aeration, very important to, you know, it's a very controlled system for, it's very sensitive for the bacteria's growth and for the performance of the system as a whole. How was it translated when it comes to you using a pilot scale system? Because in that case, it's a lot of factors that it's going to be really hard to control, in my opinion. I worked on microbial columns, and in pilot scale, it's entirely different to what you do in lab scale, which is a very controlled version. I would like to hear your comments on that.
Yes. Although I just started operating and I operated for around a month for the pilot system, I can feel it's different from the lab scale, and it's not easy to control everything. But we developed this compact system with our investors. So I'm still figuring out how to effectively control the pilot system. But some like DO or some like conductivity meters are already included in the system. So it automatically measures every second. So I can see the correct and precise data from the system. So I think I can control it. I cannot just get used to the operation of pilot system soon, I think.
Okay, but I mean, I don't know how smooth this is going to go to, but it will be an interesting study to really understand how it's being translated from lab scale to pilot scale and what are the challenges that you're facing with that, which I hope you also can capture into a journal publication, so at least it can benefit a lot of researchers as well. Yeah, so that are my comments, and yeah, I think your outputs and your progress well in place. So all the best, and to the principal supervisor, Professor Sean, for your comments.
If I jump in, do you have any question to Wanja?
Oh yeah, I have some question if I allowed.
Yes, please go
Oh
ahead.
yeah. So yeah, it was a very interesting research, I mean the dissertation topics, and I want to ask some question for the chapter 4 and chapter 6. So can you go back to the chapter 4? Oh, by the way, I'm just visiting the UTS right now, and it's a great opportunity to attend this like seminar. And I see that like there is a point for temperature 10 and 20 and 30, but do you think that the maximum point is between 10 to 20 or 20 to 30? Like, because like, I think the temperature differences is quite big, so I don't know which one is the kind of maximum point. So do you have any idea about that?
Yes. So maybe the maximum nitrification rate will be between 20 degrees and 30 degrees. So maybe in the future study, I can do the more specific, like 22, 24, 26. I can test
Oh,
like
yeah.
other
But what
lives.
makes you think so? Like, why between the 20 to 30?
It can be from 10 degrees to 20 degrees. But from I think it can be from 10 degrees to 20 degrees. Sorry, my bad. Because as you can see in the nitrification rate between 10 degrees and 20 degrees, in the 10 degrees nitrite accumulation didn't happen because I'm assuming AOB and NOB can be balanced well, and it's just the low temperature affects the inactive bacterial activities. So it's better to see between 10 degrees and 20 degrees.
Oh, yeah.
For maybe future studies, I can be more specifically control the temperatures of the reactors and then see how it affects.
Yeah, I think you don't need many experiments, but at least such experiment can help you to understand this phenomena more.
Yes.
And then another question is, if we decrease the temperature from 30 to 10, do you expect the similar result will be obtained like this? You are now increasing the 10 Celsius to 30 Celsius degree, right?
Yes.
So what about the case inverse? Yes.
It's I think it's the matter of activity of bacteria. So in 30 degrees, bacteria activity was a little bit inactivated, and nitrite accumulation happens in the reactor. So that nitrite accumulation, and that's gonna be that's affect the MBR's operation or vector. So if in the 30 degrees, if nitrite accumulated, and then if I decrease to 10 degrees, it will definitely affect the system because of the different concentration of nitrogen species. But if it's in the stable operation, even in 30 degrees, then temperature variation will not affect the system. I mean, it will go back to this nitrification rate of 10 degrees, I believe. But that's also another in my initial plan, how to see the difference of 10 degrees, 30 degrees, and 10 degrees, because in like summer and winter, in seasons, temperature varies time to time. But in this study, I only measured the 10 degrees to 30 degrees.
Yeah, I think you already know some answers, so it could be more meaningful research if you could like add more experiments. And then I want to ask you about the chapter 6 very briefly. Yeah, so you mentioned about the energy consumption of the MBR and MABR, and you imagine the MBR's energy demand is high and MABR is lower, but do you have any kind of like quantitative data or existing like some material to support your idea?
I'm now at the initial stage of planning, so I don't have personal data to support this my perspectives, but there are many previous studies that supported this MBR and MABR energy consumption. So MBR, in the MBR system, the bubble diffuser has large bubbles than this MABR system. So normally the air goes up and the 10 to 15% of oxygen is only used in the system. But in the MABR, it diffuses to, you can't really see the bubble. The oxygen is diffused to the membrane. So with the same amount of air, the more oxygen is used to the bacterial communities, bacterial activity. So that's why energy can be saved and energy consumption is low.
Oh, so if it is, so in this experiment, which factor will you compare for the urine nitrification only or energy consumption as well?
Firstly, I'd like to see how we can effectively control the biofilm thickness and how the urine can affect this biofilm activity of aerobic nitrifying bacteria. And then maybe energy consumption can be also calculated in the future.
Yeah, because the energy consumption of the different system can be compared not in fair levels. So that's my concern. But so you need to have a clear idea what to compare in the same level. So that makes this chapter more concrete and have a more better way for the publication, I believe. So I think that's my opinion in overall. Thank you.
Thank you.
Thank you for your comments, Dr. Kim. To Professor Sean for your final comments, the principal supervisor.
Yes, question for chapter 4 and chapter 6.
Professor Sean, we can't hear you very clearly. Is it possible to speak a bit louder?
Can you hear me?
Oh, yes, great. Yeah.
So I like to focus on my question to chapter 5. So you
I think
identify.
Sorry, I think it's still quite low. Is there maybe your microphone is something not working?
Can you hear me better?
Oh, that was reversed. That was good now.
Okay, it's a reverse way. Okay, so you identify the microbial communities here. So you mentioned about Aeobacteria nitrococcus, Aenobacteria nitrospira. So what's the implication of this compared to wastewater nitrification, any other nitrification in the nature? So what did you find? Because you didn't explain detail. Can you please share?
Yes.
Detail?
So in the normal wastewater treatment plants MBR in the sludge, common AOB is nitrosomonas and maybe other nitrosococcus. And NOB, nitrospira or nitrobacter is common AOB and NOB genus. But from my nitrifying bacteria analysis, the AOB was nitrosococcaceae, which is quite rare, and NOB was nitrospira. Nitrosococcaceae was enriched as a main predominant AOB in this system because of high salinity than wastewater. So from literature review, this nitrosococcaceae was dominant AOB in the marine system or quite relatively high salinity wastewater system. So I think that's why in my system, this AOB was predominant genus. And for NOB, the nitrospira was predominant NOB rather than nitrobacter, because nitrobacter it's more likely to predominant in the high nitrite concentration condition. So in my system, maybe in the stable operation condition, I had a low nitrite concentration, only nitrate, ammonia. So that's why nitrospira was predominant than nitrobacter. Yeah, that was the novel result in my system.
So do you believe if we inoculate this bacteria in your ambient system, so our inhibition factor of retention time because of nitrite to nitrate, which means anaerobic bacteria is more required for your system to reduce the HLT. So if you put the nitrospira, more bacteria, do you think that you can improve your reactor system together with AOB, nitrococcus? So do you think that this will increase your reaction for the nitrification?
Yeah, if we can selectively collect this AOB genus and NOB, if we add a lot of NOB and NOB in the system, I think we can more stabilize this system quickly and maybe increase the nitrification activity. But yeah.
Can you buy this bacteria and then add in your bioreactor manually more, and whether you can improve more?
Yes, I'll have a look if I can buy this specific bacteria in the market, and maybe I can plan that kind of study as well.
Also, you mentioned about high salinity has more nitrococcus and that nitrospira, so that you can buy from seawater nitrification bacteria solution from aquarium, whether you can improve your system.
Yes, I'll have a look and search if I can buy this bacteria from marine aquarium and increase the nitrification.
Yes, because this is the important information, and you have to utilize this data for your next experiment.
Yeah. Okay.
Otherwise, yeah, I'm fine with your progress. Thank you.
Thank you.
All right. So there's no further comments. So Winjung, we're going to put you into a waiting room, and the panel's going to have a discussion on your assessment, and I will bring you back. So just stay with us.
Maybe shall I go out for your internet meeting?
Yeah, I think yeah, it may not be relevant for you.
Okay, sure.
So we can see you the other Zoom
Okay, thank you.
meeting. Yeah, see you, Dr. Kim.
See
Bye.
you. Bye-bye.
Gayatri, I think it's better to stop the recording at this point.
Right.
Yeah.
Yeah.